Okay, so we are going to talk about enzymes. This is chapter 8 on enzymes. So what an enzyme is it's a protein that has what's called catalytic activity. So actually all enzymes are proteins except for one, which we mentioned a while ago. The ribosome actually acts like an enzyme, which is made up of nucleic acid, made up of rRNA. So that's the only example of an enzyme that is not a protein made up of amino acids. So what an enzyme is it's a protein with catalytic activity. So a catalyst, you'll remember from general chemistry, a catalyst is something that increases the rate of the reaction. It's not consumed in the reaction, and it doesn't change the overall free energy of the reaction. So let's just draw an energy diagram here to remember what a catalyst is. So this would be, we'll say, the free energy delta G, or just G, I guess, just the free energy. And this would be the progress of the reaction over time. So let's look at an uncatalyzed reaction. We'll say here is the initial free energy. So this, we'll say green, is no catalyst. So this, at where we start, this would represent the reactants, and then down here, this would represent the products, and then this difference here from reactants to products, let's say here to here, this would be the change in free energy, this difference here. So now let's look at a catalyzed reaction. So you start out with the same, that's the same place, and we're going to end at the same place. But what happens is adding a catalyst reduces what's called the activation energy. And the activation energy is the size of this hill. These should end at the same place. The activation energy is kind of this hill that is climbed from where the initial free energy of the reactants was to the top of this hill. So you'll see what happens is with a catalyst, we've decreased the activation energy. So what's true is adding a catalyst increases the rate of a reaction by decreasing the activation energy, which is abbreviated E sub A. So you'll notice that the overall free energy is not changed. So this is all true for any catalyst. So in biochemistry, we're talking about chemical reactions in living cells. All chemical reactions in our cells are catalyzed by enzymes. So what these enzymes are doing, adding a catalyst, is increasing the rate of the reaction. It is not changing the overall free energy of the reaction. So remember we talked about exergonic and endergonic reactions and how we couple them sometimes to drive a non-spontaneous, non-favorable reaction. Adding an enzyme does not take an unfavorable reaction and make it favorable. Adding a catalyst or an enzyme only increases the rate of a reaction that was already going to happen anyways. So these enzymes we said are proteins. So everything that we know about proteins, primary, secondary, tertiary, quaternary structures, everything we know about a protein is also true for a catalyst, for an enzyme, sorry. So we're going to talk about some specific examples of enzymes and roles of enzymes. There's a good slide in the PowerPoint lecture. There's lots of examples, but there's one slide, slide number 9 in your PowerPoint on chapter 8, talks about enzymatic rate enhancement. And what it does is it shows by what rate these enzymes are increasing the rate of these catalyzed reactions. So one that we've talked about before is CAN on your little chart on slide 9 of the PowerPoint, which is CAN stands for carbonic anhydrase. Carbonic anhydrase is one of the enzymes that is responsible in the carbonic acid buffering system that changes carbon dioxide into the bicarbonate ion that can be transported in our blood. So like hemoglobin transports oxygen, carbon dioxide does not have a transport molecule, but what it does have is a buffering system to convert carbon dioxide into a soluble bicarbonate ion, and then in the lungs it gets changed back to carbon dioxide and exhaled every time we breathe. So this enzyme carbonic anhydrase, if you look at this slide number 9, CAN, carbonic anhydrase. This is involved in the carbonic acid buffering system. And again, this is on slide number 9. It shows that carbonic anhydrase increases the rate of this reaction 10 to the 6th seconds. So it increases it on a scale of a billion times. So all of our chemical reactions that are energetically favorable would happen eventually, but we would die while we were waiting for them to happen. So this is increasing the rate of this reaction 1 million times. I don't know if I said billion. 1 times 10 to the 6th, a million times. So we need these enzymes to increase the rate of our chemical reactions. They don't change anything about the free energy, they just help it happen more quickly. One way that you'll know we're dealing with an enzyme is because enzymes are usually named for the chemical reaction that they are involved in, and enzymes will usually have an ASE, an ACE suffix, which tells you what kind of reaction is happening. So let's just look at an example. So for example, in a few weeks, we'll start talking about glycolysis. The first step in glycolysis is taking glucose to glucose 6-phosphate. And this is catalyzed by an enzyme called hexokinase. And you know this is an enzyme, hexokinase, because it has this ACE suffix at the end. That means this is an enzyme. Hexo means 6. This is a 6-carbon sugar. Kinase is a special kind of enzyme that's either adding or removing phosphate groups. And we usually will see enzymes written over the arrow, meaning this is the chemical reaction that's happening, glucose to glucose 6-phosphate, and the enzyme that is facilitating this reaction to happen more quickly is written over the top, hexokinase. So hexo, 6, kinase. In this case, we are adding a phosphate. We happen to be adding it to the 6th carbon. Glucose 6-phosphate means we took a phosphate group, we added it to a 6-carbon sugar glucose. The position we added it to was the 6th carbon. So that's an example of how you'll see enzymes written in chemical reactions. So before we get into a specific example of a type of enzyme, let's just talk about a few more characteristics of enzymes. Enzymes typically require cofactors. And a cofactor can be one of two things. It can either be a metal ion, or it can be an organic molecule. Cofactors that are organic molecules are usually called coenzymes. Another word we use for coenzymes are vitamins. So all of the vitamins, A, B, C, D, E, K, those are all coenzymes, and those are all important in enzymes. They are a cofactor because it's an organic molecule. More specifically, it is a coenzyme, and those are our vitamins. So we'll talk more specifically about examples of those later. One metal ion that I want to bring your attention to is magnesium. So any reaction that is involving ATP is going to require the metal ion magnesium. So ATP any enzyme involving ATP, so ATP enzymes will require a magnesium cofactor. So you'll see that. You'll see that written sometimes over the arrow. You'll see Mg2+. The reason for that is because ATP, adenosine triphosphate, has these 3 phosphate groups on it. Those phosphate groups are all negative, and it is the ATP is going to be stabilized in the active site of that enzyme by this positive magnesium ion. You'll also see zinc ions as cofactors. This is the one we're going to see the most of, and we'll talk about this a lot more once we get into glycolysis in a few weeks, but just keep a heads up for that. Another thing to talk about is that many enzymes are synthesized in an inactive form. So I'm going to put this over here. So many enzymes are synthesized in an inactive form. This inactive form is called a zymogen. So a zymogen is an inactive form of an enzyme, and something needs to happen to this zymogen for it to become activated. So this makes sense if you think about it. So enzymes are just proteins. So when we're making these enzymes and we're making this protein, we might not want it to be active right away, because once an enzyme is active, it's going to go around catalyzing whatever reactions it is supposed to catalyze. So let's just look at an example of this. One example is a digestive enzyme in our stomachs called pepsin. So pepsin is not synthesized as active pepsin. It's synthesized as something called pepsinogen. So pepsinogen is the inactive form of pepsin. And what happens is when pepsinogen reaches the part of our stomach where it is ready to break apart proteins, And HCl, hydrochloric acid, which is in our stomach, the hydrochloric acid is going to cleave off the inactive part of this enzyme, and that will produce active pepsin. So this pepsinogen is inactive, and then when it's ready, when there's HCl present, it cleaves off the inactive portion to form active pepsin. So this happens with a lot of enzymes where they're synthesized in an inactive form, and then they'll be modified in some way to then become active. So enzymes are highly specific. So just like when we were talking about hemoglobin, and we said oxygen was binding at the binding site of hemoglobin. Hemoglobin is not an enzyme. Hemoglobin is a binding protein because it's not catalyzing a reaction. It's binding oxygen, but it's not catalyzing any sort of chemical reaction. It's just transporting oxygen from one place to another. So don't get confused about binding proteins and enzymes. For an enzyme to be an enzyme, it has to be catalyzing a chemical reaction. But similarly to the binding site of hemoglobin and its attraction for oxygen and how that can be changed, those conformational changes with the cooperative binding, we also have that in what's called the active site of the enzyme. So the active site of the enzyme is where what is called the substrate binds. So substrate might be a new word for you. Substrate is just what we would call the reactant. If this was a chemistry class, we would say these are the reactants and these are the products. In biochemistry, we call the reactant the substrate. So over here with my example of glucose to glucose 6-phosphate with this enzyme, hexokinase, glucose would be the substrate. Glucose 6-phosphate would be the product. So the active site is where the substrate binds. So let's just draw an example of that. So let's say, and there is a real picture of this in your slides on slide number 14. Let's just say here is hexokinase. And hexokinase has a specific binding site for glucose, and we'll say it's right here. We'll say this is the glucose binding site. And it's got a perfect little puzzle piece site in there where only glucose will fit in. It's kind of like a lock and a key, the specificity between the substrate and the enzyme. And what happens is after the glucose comes in, the whole shape of this hexokinase changes. I didn't draw that very well, but it kind of closes around that glucose. And you'll see this on your slide, on slide 14. But it causes a conformational change, and this is where the actual chemistry is happening. So there also would be an ATP binding site in here, because this is a specific example where we're adding a phosphate group. We're going to add a phosphate group from ATP, but it's going to happen in here in the active site of this enzyme. And you don't need to know anything specific about the mechanism of this reaction. We'll talk about this when we get to glycolysis. This is just an example. But there's highly specific interactions between the active site of the enzyme and the substrate that binds it. So these are all going to be non-covalent interactions. And these non-covalent interactions are going to be between the R groups. between the R groups of the amino acids in the enzyme and the substrate. So this, we said, this is the active site. The active site is where the substrate binds. So many enzymes will actually have other sites. Any site other than the active site is called an allosteric site. So for example, if there was any other place here, any place that isn't the active site is called an allosteric site. And this is where anything other than the substrate binds, so not substrate. And just like with hemoglobin, even though hemoglobin was not an enzyme, it was a binding protein, but we also saw the allosteric sites of hemoglobin where the affinity for oxygen could be affected. We saw 2,3-BPG. We saw a few other places where things that were not oxygen, not the substrate, even though this was not an enzyme, just drawing some parallels here. Anything that's not a substrate, if it binds to the enzyme and can increase or decrease the affinity of the substrate for the enzyme, that is called the allosteric site. Okay.